Everyone, and welcome again. And this session today is about the cell cycle, and as you can see there with my title, I actually include interphase, mitosis and cytokinesis. Okay, so we'll talk about all three of those processes. Okay, so just to familiarise yourself, if you're not familiar with the format for the webinar, I'm sure you are by now, but remember it's 40 minutes long and there is a facility for you to ask questions, and I will leave some short pauses for you to attempt the questions. They're shorter than I normally would do, but just to get through the work in the time. But of course, the idea is that the materials will be sent out to you. You'll see the URL, and then you'll be able to go back over this work if there's anything that maybe I was unclear about, or that you want to sort of go back over it again to revise at a pace to suit you. It's all here for you then. Okay, so let's get on with what we want to talk about today. So as I say, it's basically three elements to today's session. Can you describe what happens in interphase? So what exactly happens in all of the parts of interphase? And then we're going to look at PMAT. So that is mitosis. So you need to get into that habit of thinking mitosis equals PMAT. And at the end of mitosis, there is something called cytokinesis, which is cell splitting. And then we're going to look at the significance of mitosis, not only in terms of why it's so important for cells, but in the context of damage and disease as well. So let's just do a quick little recap, because you'll be familiar, I'm sure, with this idea that different species will have different chromosome numbers. But in mitosis, what happens is that you produce cells that are exactly the same number. So as humans, you know, we have chromosome number 46, we will produce cells, daughter cells, in mitosis that are genetically identical and of that number. And then of course there you can see, you know, if we were looking at chimpanzees, then it would be a different number, and that's what makes a chimpanzee, not a human. So you can also see in the images I've put on there that you're also familiar with the idea that chromosomes are found in the nuclei, and that when chromosomes make a copy of themselves, when the DNA within them replicates, then you will see those structures as not just the one chromosome, but as two chromatids. Okay, so and the total number of chromosomes in a cell in a body cell is called the diploid number, and gametes will have half that number and they're called haploid. When you see a chromosome, as you can see in this sort of middle diagram at the top, you're made of two strands. Each strand is a chromatid, but the one structure is a chromosome. All right, so that is one chromosome, and you can see in all of this photograph here, all of these structures, they may be made of two strands, but they are one chromosome. Okay, moving on. So you've got to be clear about these two phases I'm talking about here, and the one is interphase, where you've got new proteins made, then you have DNA replicating, and then you've also got organelles growing or dividing, and I'll come to sort of distinctions about that in a little bit, as opposed to mitosis, which is, you could consider it as nuclear division, and those are the four phases there, PMAT. Okay, so different things happen in interphase, and it is separate to mitosis. So if we have a look at the cell cycle as a whole, then you can see a large part of a cell cycle is taken up by interphase. And you can see there the three phases I told you about, the G1, the S and the G2. And if you look at S, DNA copied there, and this is the basis of a lot of questions, that this is where the DNA in a cell is doubled, okay, because this is where DNA replicates. So you see the one structure then becoming the two chromatids in terms of a chromosome, okay, as that DNA is copied, as we've discussed in earlier sessions, and forming chromatids. And then what happens is that DNA content that's doubled between G1 and S then stays at double all the way through G2 and mitosis, so through PMAT. And what will happen then is that at the end of mitosis, so, you know, in cytokinesis here, when the cell splits, what happens then is that the DNA content in each of the daughter cells is half of what the one cell was that they were made from, if you like, the parent cell. So that is why DNA needs to double in order to make these genetically identical cloned daughter cells compared to the original. So remember, interphase not part of mitosis, and I said I'd say a little bit more about the detail of what happens in interphase. Well, the DNA is going to replicate, and I think I've mentioned that. That's where you see chromosomes each chromosome being composed of like 2 strands called chromatids. Organelles replicate. Now, I need to actually qualify that because there are only organelles that have got DNA in them, like mitochondria and chloroplasts, we describe as replicating. So it's fine to say mitochondria replicate or chloroplasts replicate during interphase. You know, it's not true to say that other organelles will replicate, okay? You can say, however, with other organelles, new organelles are made. Okay, so new Golgi body or endoplasmic reticulum is made. The other thing that happens is that we produce ATP and proteins and ribosomal material that goes to form ribosomes. So that happens in our cells and in organism cells during interphase as well. And we also say that cells increase in size, but we don't say there's growth because we associate growth with cells actually doubling in number. Okay, so you'll see interphase as the longest cycle there. You'll see there that in the images there, that little drawing, you'll see most of the cells there have got cells which are in interphase. So that's where the nucleus looks a little bit like it is when we generally see it in cell drawings. You know, you'll see it there. So when you look at a photograph, when you look at a microscope slide, you can expect to see most of the slides in interphase because that is the longest phase. So, you know, there's more of a chance of you seeing them. Now, as I said also that the DNA replicates during interphase, and you can see here, if we look at the DNA content of a cell, and we're using something called arbitrary units, then actually what happens is that during interphase, or during that G1 phase, you know, you'll have the DNA content, let's just say, at 1 arbitrary unit. And then in the S phase, what happens is DNA replicates, and you can see it will double. So this is during the S1 phase, the DNA replication will be completed. And then it stays at 2 arbitrary units all the way through the rest of S1 and into G2 and into mitosis. And then it's only when the cell then splits, the cytoplasm splits, that we actually halve the DNA. Okay, so just going back then to why it is that it doubles because, as you can see this image over here, the DNA replication produces now double the DNA, and each chromosome is now seen as 2 chromatids. So let's look at the detail of PMAT then. So we can see here the idea is that we have diploid cells producing genetically identical diploid cells of exactly the same chromosome number. All the alleles are absolutely identical. So we say that we get 2 nuclei from one, and of course in doing so we're maintaining that chromosome number. So if we're chimpanzees, we're maintaining the 48 diploid number. Now, most animal cells will carry out mitosis, so most cells in the body will carry out mitosis, as you know, maybe for growth or repair or replacement, but mitosis only occurs in specialist parts of the plant. And I've shown you a little image here of a growing tip, a shoot tip of a plant, and you'll see that there's sort of some leaves at the side, and you've got the growing tip there. It's called the apical meristem. Apex meaning tip, you know, a top, and the meristem is the growing part. So that's where mitosis will occur, and it'll occur in other places like the root tips as well, in the new buds, and also you'll see it in tree rings, at the sort of the cambium between the xylem and phloem. You'll see that can grow mitotically as well. So that's what forms sort of circular tree rings. So you need to be able to explain what happens in PMAT at each of the phases, and to be able to recognise them both in a diagram and also from images as well. So this is what we're about to do. So if we look at prophase, so I've got a few images here. So you'll see this diagram in the middle here. You can see separate chromosomes and they look very sort of ordered there, and you very often don't see it like that because don't forget, each of these phases is a continuum. It's not just when you see a cell in prophase, it always is in the same stage of things happen. Things gradually happen during prophase. So what happens is the DNA condenses because the chromosomes are just very long and they're very thin, and they get shorter and thicker, and that's when they become more visible. So you'll see in prophase, this image on the right hand side here, you can see the nucleus doesn't look like it would normally look like in interphase. There seems to be strands there. Okay, so that is how you'll recognise that as maybe prophase. So the chromatids and certainly chromosomes will become more visible. Now, in an animal cell, you will also notice, as you can see this middle diagram here, that there are centrioles, and they will move to opposite poles of the cell, and they will begin to start developing a spindle. So this is where it begins. You'll see later on in prophase, so you can see that this is sort of, you know, more of a middle prophase here, whereas this diagram over here is a late prophase because the nuclear membrane has completely broken down. And as I say, this is a continuum throughout prophase. The nucleolus, which you'll also see as like a spherical, very dark object in the nucleus, that will have disappeared as well. And so by the end of prophase, you'll see pairs of chromatids in the cytoplasm, you know, taking up quite a lot of the cytoplasm. So the next phase, metaphase, then, I always think M for metaphase, M for meeting in the middle, and you'll see your chromosomes. What happens now is the spindle is developed, and you'll see the chromosomes there aligned along the equator, and you'll see they're at right angles to the poles. Okay, a couple of images here for you to look at, but they don't always appear as tidy again as the diagram. Don't forget that is a diagram. And so over on the right hand side here, I've shown you a little image. We've got a plant cell here. There we go. It's a rectangle I'm drawing around now for you. And the chromosomes are arranged in the middle, in this metaphase where they're meeting in the middle. So you can see in the diagram, but you can't see it in the image, I know, that the chromosomes will become attached to the spindle, but they're becoming attached by their centromere. Okay, so that's important for you to remember. And then what begins to happen in metaphase is the spindle fibers, which are made of this protein. I'll say a little bit more about that later. What happens is that begins to contract, that protein, and so as it contracts, it pulls the chromatids apart from each other. Okay, so that is what is just about to begin to happen in very, very late metaphase. So in anaphase, then, we can see now that actually the centromere has split. So we've now got two parts of the centromere there. And what's happening is that those spindle fibers are contracting and they're shortening and they are pulling the chromosomes, so that it's leading by the centromere all the way to the poles. And you can see how it is in this diagram on the left here, how it is that the centromere pulls. The chromatids are being led by the centromere and they're heading towards the pole. And you can see here that this diagram is anaphase. The chromatids are separated from each other and they're heading in the opposite direction, is how I would recognise anaphase. Telophase, some people say telophase, some people say telophase, and what you can see here is this is the final stage of PMAT, of mitosis, of nuclear division, and the chromosomes will have reached the poles. They're now the chromatids that were being pulled apart will have now reached the poles, I should say, and so they're now known as chromosomes. Okay, they will eventually then, during this phase, uncoil again. They'll get very long and thin again, and you won't be able to see them in the detail that we've seen before. The spindle will have broken down, it'll have disappeared, and again the nuclear membrane will begin to form over around the chromosomes, and a nucleolus will again become apparent. Okay, so that's telophase. So we've now made 2 parts to the cell, in actual fact, which are now genetically identical. And of course, you can begin to see something happening in this image here, where the cell seems to be sort of pinching in a little bit. And that is something known as cytokinesis, which I will come to. But I just mentioned earlier on about the protein, because I wanted to show you some images of the protein, the tubulin protein that forms the spindle, and you can see it very often when you see slides. You can see it stained, as you can see. It's like a you know, quite dark pinky red colour here. So that's this is the protein spindle, and you can see the chromosomes are very dark blue. Okay, so you sometimes see, like, it doesn't look neat and tidy like you'd see in a diagram. It looks like a bit of a mess. So you know, make certain you're actually looking at chromosomes and not spindle. And this here, you can see it's shown up as fluorescing in this case. So, but this is the sort of the tubulin, the protein fibers. Okay, so need to be careful to sort of, it gives you an idea though of where the chromosomes are, but be careful you're looking at chromosomes as opposed to spindle. So what I thought we'd do now is we'd have a little go to see if we could recognise these different stages in the cell cycle in PMAT, because as I've said, this is one of the strategies that you need to be able to put into place. And this is a little recap of a number of different images here. And I've shown you this because I want to show you there's subtle differences between interphase and prophase when you see them in microscope images, because the nucleus here is very much darker, and as opposed to prophase, you can't really see the outline of a nuclear membrane, and this definitely seems to be more a sort of stranded appearance there, if you like. And down here again, you can see in interphase, you can see there's a very dark nucleolus there, but in prophase here you see that there's definitely more of a stranded appearance. So that's the one that people have most difficulty with generally, because if the chromosomes are in the middle, most people are happy to say metaphase. Anaphase, they've moved away from the middle and the chromatids are heading to the poles, and by telophase or telophase you can see the chromosomes, as they're now known, are at the 2 poles. So okay, here we go then. I'm going to show you some images, and I'm just going to give you a couple of seconds for you to decide PM or T. So here we go, have a think, and it is metaphase. Absolutely right, because you're meeting in the middle in both of those cells. Next cell then, so decide, is it PM or T? So 3, 2, 1, and it is prophase, absolutely, because you can see the strands there. Next one, so we have this cell that I've highlighted in the red box there. So 3, 2, 1, yes, it's anaphase, as I'm sure you've actually correctly identified. And let's look at this one, and you can see there the chromosomes have actually been separated, the chromatids have been separated. They now look like they are chromosomes, anaphase. Okay, so you know, just be careful that actually what we've got there is we're actually not far enough for that to be telophase. Okay, and then let's look at the next one. I've got 4 slides here, different phases. What would you actually say each of these phases are? Okay, so take a bit of time to look. Okay, so here we go. I'm going to go through them. So this one at the top left is prophase. So you can see, you can't see a nuclear membrane. If this is the outside of the cell, you can't see a nuclear membrane. You can see though that the chromosomes look like they're different structures. You know, they're not long and thin, they're visible as separate structures. Next, anaphase, because you can see that we're definitely not meeting in the middle, they're heading towards the poles. So the chromosomes are anaphase. Let's look at this one over here. That is telophase, because you can just see maybe that the nuclear membrane has just formed, and so the chromosomes are now at the poles surrounded by the nuclear membrane. And this one's a strange one, isn't it? Because we're expecting to see them in the middle of the cell, but it might just be the way that that cell has been sectioned for sort of observation under the microscope, but they are meeting. You can see chromosomes here meeting, and that is why that is metaphase. So Cytokinesis then, as I've said, this is literally cytoplasm splitting, and this is the phase between, it's sort of after PMAT as far as mitosis is concerned, and it's before interphase starts again, so that's before interphase G1. Okay, now it's different in plant and animal cells though, and this is something worth remembering. Okay, so we discussed earlier on, you've seen that graph that mitosis is C, so we've got during mitosis, we've still got our double DNA in each cell, and in cytokinesis, in D there, what happens is the DNA content halves, because half goes to one daughter cell and half goes to the other. Now, I mentioned that animal and cells look, they carry out cytokinesis in different ways, and they look different. And in animal cells, we've got this kind of furrow, we've got this nipping in. So what happens is a kind of constriction begins at the outside of the cell and gradually pinches and travels inwards. And that is what animal cells look like in cytokinesis. Plant cells, however, what happens there, look at this diagram here, you can see cytokinesis begins in the middle of the sort of the inside of the cell, where something called a cell plate begins to form. Okay, so that's where the equator was, where the chromosomes met in metaphase. And so what eventually happens is that, you know, cellulose will be laid down along that cell plate there, and that's what eventually will lead to the two new cells. So it's slightly different, even though cytokinesis occurs in plant and animal cells, it's slightly different between the two. You know, just a diagram there just to summarize that process. Okay, so the next part of the cell cycle I wanted to talk about is related to the significance of mitosis. And we know, as you can see in the bottom here, I've talked about growth, repair and replacement. So we know some cells will die naturally, you know, after a certain period of time, they need to be replaced. Others need to be repaired, and we also know that an increase in cell number leads to growth, and we know that is sort of in meristems in plant and generally in just about any cell in animal cells. So that's the one function of mitosis. The next is that it maintains genetic stability. So as I've said, in mitosis, we'll produce daughter cells from the parent cell that are genetically identical. They're clones, they've got the same number of chromosomes, the alleles will be the same. And then the last part is asexual reproduction, because some organisms use mitosis as their method of reproduction. And you can see here with some of these images, like bacteria, for instance, when they actually undergo mitosis, we call it binary fission in bacteria, they'll produce two cells. Again, amoeba, that's a protoctistan. I don't know whether you remember that or you've seen that before. And we've got an amoeban which is in the process here of producing two daughter cells, one along the top there and one down here. So that's amoeba. Okay, but don't also forget that even though plants can reproduce sexually, there are times when actually, in order to survive, maybe the conditions are not suitable for sexual reproduction, that they'll produce clones of themselves in asexual reproduction. And we've got a spider plant here, and it's called Tradescantia. And what happens is cells will run down through these tubes that are coming off the parent plant that you might be able to see there. And then what will happen is that they'll start growing by mitosis and produce genetically identical clone plants. But of course, they're close to the parent plant, which is not such a good idea for survival. And of course, you've got no genetic variation, which again can impact potentially on survival. So that is the sort of the one significance of mutation of mitosis. Sorry. The other thing I want to talk about is mutations in the DNA, and I'm talking about now in the DNA of the genes that are controlling the cell cycle. Okay, because what can happen is that, as you can see in this diagram on the right hand side, you can sometimes have mistakes occurring during the copying process of the gene, during the replication process. And of course, if those mutations occur in genes that are controlling the cell cycle, then maybe the cell will divide too rapidly, maybe it won't divide at all, maybe it'll divide at the wrong time. And then ultimately what can happen is you might have this uncontrolled cell growth, which leads to a mutation, a tumour forming. What sort of things cause these mistakes or cause these mutations? Well, it's things like radiation, viruses, certain chemicals. They can all cause mutations. Yeah. So as I've said before, these genes controlling the cell cycle, they normally they act as a natural break between mitosis finishing and then G1 in interphase starting again. But if there's a mutation in those control cells, then you have this uncontrolled growth, as I've said. And of course, some genes have the potential to actually change to an oncogene, and that's a gene that will cause cancer. Now, I wanted now to link that with the fact that there are some drugs, because this has been a question, exam questions that have come up as well before now. So there are drugs that will block some part of the cell cycle to try and limit the growth of that sort of uncontrolled growth. So cisplatin, methotrexate, vinca alkaloids like vinblastine, vincristine, vinblastine, you know, a brilliant drug in the treatment of leukemia. Just it was first discovered, I believe, in the rosy periwinkle in the sort of the rainforests. Really, you know, ordinary looking plant that held, you know, and holds so much promise for medicine. Doxorubicin as well. So cisplatin and methotrexate prevents DNA replication. So what you want to be thinking about is how this will affect the part of the cell cycle. So if we can't replicate DNA, then that's the S phase in interphase stopped. If we interfere with spindle formation, as these drugs do here, then actually we can't have metaphase. So again, it'll interfere with the cell cycle. And then the last drug here will target enzymes, essential again for the cell cycle. So they target in different parts of the cell, and in an attempt to actually block the uncontrolled growth. So let's put some of that into practice then now, because what we can see here is a diagram very, you know, similar in a way to what I showed you earlier on, which is interphase occupying most of the cell cycle there, and then also some other phases as well. So what we've got there is PMAT. Okay, and it says draw an arrow on the diagram to show the correct sequence of events. So this is wanting you to work out that PMAT occurs in a certain order, and it's no surprise to us, is it, that actually it's in that order, because that's how we learned it, PMAT. Okay, so draw an arrow nice and clear on the diagram there to show that sequence of events. And then it says add a segment to show the point at which cell division or cytokinesis would occur. So from what I've said, can you remember where it is that cytokinesis occurs? Where would you put it? How big would you make it? Okay, so here we go. There we go. So it's just after PMAT, and I've kept the little slice of pie there about the same as say telophase, so that you know it is about that sort of same length. And then for events that occur during interphase, well, all of the things I talked about, you know, the DNA replication, the replication of organelles, some organelles made, ribosomal material, ATP, you know, there's a whole host of them there that if you could learn them, it is quite a common question, you know, what happens during interphase. So there they are for you. Okay, so that's one question. Oh yes, I just thought it was worth just mentioning again. Remember, you know, we're not talking about cells growing, you know, because if we're talking about growing, we're implying, you know, something that is not happening here in interphase. And I've already mentioned that as well, because replication is just referring to mitochondria and chloroplasts that have their own DNA. Okay, so there we are. Moving on. So, all right then now, so I'm just going to go through this. Name the stage of mitosis where each of the following occurs. So what we've got here is chromatids lining up at the equator. So see if you're right. I'm sure you were. It's metaphase. So if I said, in which phase do centromeres split? Okay, so that's something maybe you need to have another little think about. So were you right? Anaphase. They have to split in order for them. Remember, they're leading the way to the poles by the centromeres, which have had to split because one centromere is attached to each chromatid. Spindle fibers contracting and shorten. So which phase? Anaphase. Hope you were correct. And then what happened? Of course, you know, they're contracting and they're shortening, and that's what actually is the sort of pulling force to take them to the poles. Chromosomes first visible as a pair of chromatids, and that is prophase. Absolutely. Remember, we talked about how it is that you could recognize prophase as opposed to interphase. Nuclear membrane reforming, and there we go. We've got telophase. Okay, so those are sort of you know, sort of hopefully sort of an easy list of questions for you. So let's move on to the next one. Now, this is a very common type of question, to actually draw something out, showing a single chromosome in this case as it appears during prophase. So if we think about that, we're prophase, so we're shorter and we're thicker. We're actually made of 2 strands and we're held by a centromere. So if you have a little look at this, this is exactly the sort of thing that you want to actually be drawing. So you can see here that we've got 1 chromatid, 2 held by the centromere. Each one is a sister chromatid. Okay, so you know, we can see the features there. Sometimes questions ask you to label them as well. So make certain you can label them there. It says a labeled diagram in this case. They don't always, but you know, just look out for that as part of the question. You don't need to colour them in, you don't need to shade them in, just a simple line drawing like that. But remember, keep each chromatid the same, keep it the same shape and the same size. And so next questions then, describe and explain what happens to chromosomes during anaphase. Well, you know, we've talked about that, haven't we? So the answer there is, we know the centromere is splitting. We know the chromatids are being pulled to the opposite poles because the spindle fibers are shortening or contracting. That tubulin protein is contracting. And then name the cell organelle responsible for the production of the spindle fibers. And that is, have a little recall, and that is centrioles. That's right. Okay, so that's another question. So you will see this appears not only in Bangor resources, which you can use for revision, but you'll see this table here appearing in several past papers and tests as well that you might have come across. And it shows us that actually what we've got here is the DNA content of a cell measured during 1 cell cycle. So we've got our phases here. So you can see we've got our interphase, we've got mitosis, and we've got cytokinesis. And it shows us how it is the DNA will increase during this S phase, which we now know is interphase. So it'll double from 20 to 40. It'll maintain that content through G2 and into mitosis. But then during cytokinesis, look, it'll halve, it'll go from 40 to 20. And that's because each of the cells, each of the daughter cells will get the half of the 40, which is 20. Okay, so if you look at it, it's the content of a cell, not the content in total, the content of a cell. So this is why we've gone from 40 to 20. So please look out for that in tests and questions that you've come across. So name the period of the cell. So I'm hoping now that you're going to get that straight away. And the answer to that is absolutely, it's interphase. And again, look, another common question, 2 events that occur during the period. Well, we've talked about those. You know, we can roll those out again and you can take your pick from any of those. Using the information provided in the diagram and the table, explain why it's important that the DNA content of the cell increases during stage S and decreases during stage C. Well, I think I've, said that at the, when I was going through the table at the top there, you've got to have double the amount of DNA and S so that the daughter cells, each daughter cell can get the correct amount of DNA and so maintain the chromosome number and the content. So the DNA is doubled during S. And again, look, I'm using data from the table, 20 to 40, so that each daughter cell now is half. Make certain that actually you are saying the obvious things as well, okay? And then how does this maintain genetic stability? Well, we've got identical clones, we've got 2 identical daughter cells produced, and that is how we maintain that stability. A very common, you saw this image at the start, but a very common sort of image where it asks you to label the cells and with the different phases. So if you look at it, A, have a look at A, have a look at B, look at the fact that you can see some strands there. If you look at C, yeah, and D, yeah, okay. And so now A, B, C and D are, there we go. So we can see that D meeting in the middle, they've not started heading towards the poles. We can see that C is telophase because we are well and truly at the poles there. Okay, and you should be okay with A being anaphase because we're heading to the poles, but we're not right there. The strands are still in the middle part of that cell to an extent. Okay, and hopefully you recognize prophase now. And the cell cycle, which stage? Can you see in greater numbers? Again, a very common question. Interphase, it's the longest phase. And I just wanted to sort of share this one, which is we've got 20 pairs there, and it says, what's the diploid number of this mouse in this question? 40. Remember, don't say 20 pairs, it's asking you for the diploid number, which is 40. And another common question, moving on from that, is it says, using the cell outline below, show how these pairs of chromosomes are arranged. Now, I mentioned that actually they're arranged on the equator at right angles to the poles. So if this is the equator, they will appear here. Okay, the original question was about meiosis, but I've adapted it for our purposes today. So it's meiosis. So you can see the pairs there. Okay, and again, please don't miss out things because you might have to label chromatid, centromere, etc. So, okay, this question is a little question about the different stages. And what I want us to do now is to think about putting them in an order. So let's think about if we had a cell cycle, which of these would be first in your cell cycle? So let's start off with B. All right, so which one do you think would come next? So we're on that for prophase. So next, sorry, B was interphase, sorry, and now D is prophase, sorry. And then the next phase after that, what are we going for? Are we going for, yes, we're going for C, which is metaphase. And then the next phase, what are we going for this time? Absolutely, F. Yes, we can see anaphase there. Next phase, yeah, we can see the nuclear membrane look beginning to reform. And finally, we're then heading then into cytokinesis. Notice it says cell cycle, not mitosis, because we've got cytokinesis beginning to occur there. And then just one last one, because you will need to recognize these. Here we go. So I'm just going to reveal them. Anaphase, B, metaphase, C, sorry, we'll do D first then, telophase, telophase, and C, prophase. Okay, and once again, I do like to sort of include a little bit of information here on a QER question. So what we've got here is the question, which is in 3 parts, the different phases of the cell cycle. How is genetic stability maintained in mitosis? What's the significance in damage and disease? And we know we'll have 3 parts to that question, which, as usual with me when I approach these questions, as you may know by now, I get all the keywords together then I put them in a sensible order, and then I can then get my sentences ready in order to link each sentence next to the other, but in a sensible order. And remember, it's all about getting your keywords accurately sequenced, and you've got all parts of that question covered with no major omissions, so you can be in the top band of marks there. Okay, if you're going to put a diagram in, remember you have to annotate. And so as usual, what I'm doing here is I'm just going through, I've put them in order for you. I've got the keywords. So what you can do as a little exercise at your own pace is you can think of the keywords, and then once you've got all the keywords together, you can then put them in order as I did then. And I'll just finish today by just reminding you that, you know, I quite like these dice sheets, but what I've done ready for next week as well is I'm leaving you with a dice sheet that's got mitosis and meiosis on it, and what you can do as part of revision for tests, etc., is you can put your answers there, and what I will do next week is I will give you the answers. Okay, so that's all from me for this session. All that remains now is for me to say thank you very much for listening. I really hope that that's been useful again. And our next catch up is next Wednesday, where we're actually bringing that all together with meiosis, and we're comparing and contrasting both processes. Okay, so I'll say goodbye for this week. Goodbye.